Good morning once again. My name is Olatunde Michael, the CEO of Omega Bio-Edutainment. This is in furtherance of the educational series of Omega Bio-Edutainment. Today, we are going to talk about, continue our discussion on the innate immune system. Innate immune system. I told you that the major characteristics of innate immune system is that it is an inborn characteristics. Then number two, it is non-specific because it is still naive. It has no prior exposure to any immunological challenge. Then apart from that, it is very in action, very quick to respond. And today, just like I said, today we are going to look at what are the basic components of the immune system, the basic component of the innate immune system. So that is, if we are to be immunologically competent at birth, what are those cells that should be there in order to ward off against infection? The first component of it is that it has a physical barrier. It has a barrier, and this barrier, this one is a physical. Example of this one is the skin, the one that is being put up by the skin. Don't forget that the skin is the first line of defense. Once the skin barrier is being broken, that will give way to the entry of a robber is able to scale the fence or come in through the main entrance. So the next thing for you is to look for a way of having to send them out or overpower them. So the same thing, the skin is the first line of defense that is being mounted by an immunologically competent host to ward off against infection. So, and if you look at the surface of the skin, you discover that it is highly keratinized. It contains keratin. It is highly keratinized because it is made up of different types of protein and dead cells. So, because of this attributes, some microorganisms are being warded off by the skin. Then the second aspect of a physical barrier is the one that is being put up by the cilia. You will see that there are some special cells like the one that is being put up at the alimentary canal by possessing cilia. So this cilia will just make sure that there is a movement, for example, peristaltic movement, that's going to send the microorganism into a place of no return. So that is that. Then the next one is chemical in nature, chemical barrier, and this is the one that is being put up by tears. You will discover that the human eye is continuously being armed with tears, water, to wipe away any kind of disturbance. So that's why you see human eye is it contains tears, water that contains some hydrolytic enzyme that will degrade some microorganism. Then saliva is another. Then the stomach contains hydrochloric acid, which make them uncomfortable for certain microorganisms to be able to grow because of the high pH. That high pH will hydrolyze enzyme, will degrade certain enzyme from the bacteria or from this. Then apart from that, we now have cellular components of the innate immune system. So this includes the one that is being put up. We are going to consider natural killer cells, which is shortened as NKC. Natural killer cells. This is we are going to see we are going to look at the structure, the components of each of these cells. So after natural killer cell, we will consider mast cells. What are they? What do they contain, in order to make sure that they prevent or they protect the host from certain pathogens, from the establishment of by certain pathogen. So thereafter, we will look at eosinophils. What are they? What do they contain, so as to be able to say? Then number 4 is basophil. Another cellular component of the innate immune system is the one that is being put up by basophil. Then the 5th one, the one that is being put up by macrophages. Macrophages are professional phagocytotic components. That is, they degrade. They have the ability to degrade by swallowing. They degrade by swallowing. Swallowing. That is what is called a phagocytotic mechanism. So after this macrophage one, the 6th one we are also going to talk about is neutrophils. Neutrophils. That is the one that is being put up by neutrophils, like leukocytes and others. We are going to look at that. Then the 7th one is we are going to consider dendritic cells. Dendritic cells. For example, the case of when you get to the lungs. So the dendritic cell will be recruited to fight against infection by that. So that is the second aspect of it. Don't forget that I said we are looking at the innate immune system, the component of the innate immune system, and we look at the physical and chemical barriers that is being put up by the host in order to be able to fight against infection. Under physical barrier, we look at skin. I told you that skin is highly keratinized, and it is made up of certain epithelial cells that will make them too inhabitable for certain microorganisms to be able to establish themselves. Also, we look at cilia. I told you that there are certain cells that are ciliated. They contain cilia, numerous hair-like structure that will prevent the establishment of microorganism in them. Then another component that is chemical in nature. I told you that these one are chemicals. That hydrolyze, that highly degrade microorganisms. They have the ability to degrade microorganisms such as tears, such as saliva. Then I told you, the one that is stomach, whereby you have hydrochloric acid as major component of the stomach, that make sure that the microorganism does not survive in that high pH. So, and another one that I did not mention is that of peroxide. So by degrading hydrogen peroxide into oxygen and water, so that is will still come under chemicals. So this second aspect, which is cellular in nature, and under this we have majorly we have seven. The first one we are going to look at is the natural killer cells, then followed by mast cells. Then we are going to look at the structure of eosinophils, basophils, macrophages. I told you that macrophages are professional phagocytes that degrade foreign compound or foreign antigen by swallowing. Then, and the last one is the dendritic cells. So we are going to look at that. So immediately after that, we will now consider the third, which consists of the complement system, which contains the complement system. Just like the name suggests, complement. This one will work to complement the efforts of macrophages. It's going to work in it. So these are series of chemicals. These are series of proteins. Proteins that are up to 27 different proteins that make up or we augment the effects of macrophages and cellular parts of innate. So now we are going to start taking them one after the other. We are going to start picking them one after the other. So, and if we are to start, we are going to start with the first major component. The first major component, which we have to do with the physical barrier. It has to do with physical barrier. Okay. Now, I told you that the skin will serve as barrier, prevent the establishment of infectious agents in the host, and that is because it has a layer that is called epithelial cells. Okay, epithelial cells. So these epithelial cells will actually interact with the environment. It will continually interact with the environment, thereby preventing the establishment of diseases or infection in the host. Okay, so the major, okay, so an example of where you have epithelial cells, you have them in the GIT, that is in gastrointestinal tract. You have the epithelial cell of a certain of, let me say generally, the respiratory tract. Okay, the respiratory tract. Then we also have them in the genitourinary, like vagina and bladder. All these, they are armed with movement of the epithelial cell that will prevent the establishment of bacteria or prevent the establishment of pathogen in the host. And just like I told you that in this epithelial cell, the epithelial cell is highly keratinized. And that one, because it is highly keratinized, is going to make sure that the microorganism does not establish itself in the host, in the host. Then also, these epithelial cells, I told you the epithelial cells, and I've given you examples of where you can have the epithelial cell. We have the gastrointestinal tract as an example. Then we have those on the surface of a respiratory tract, as well as genitourinary, and I mentioned vagina as an example of where the skin, the epithelial cell, is serving as preventive mechanism for the bacteria or microorganism to be able to establish itself in that. So apart from the epithelial cell being highly keratinized, it has also contained lymphocytes. It contains lymphocytes, especially T lymphocytes. We are going to see this as we go on. Don't forget that there must be cooperation between innate immune system and the other arm of the immune system, which is the adaptive system. It is at this point that there will be a kind of cooperation that the adaptive system the adaptive immune system will now collaborate with this one in order to make sure form what is called the gap junction. It also serves as the gap junctions. The gap junction, what gap junction does is that the gap junction will serve as medium of signal transduction. That is, it can also form a, a way, or it can also form a medium whereby there will be communication between two cells. It forms the gap junctions. Okay, then it also has the ability, the epithelial cell has the ability to secrete mucous membrane. And don't forget the advantages of mucus. The mucus is the one that will make sure that it provides a slimy surface, slimy, or a slippery, so make them to sleep. To sleep is just like when some on the surface you now have somebody that has poured in some slippery objects or slippery liquid on the surface of tiles. You see that everybody is going to sleep. So the part of the physical barrier that also will make sure that microorganisms do not establish themselves in the host is by secretion of a mucous membrane that will make the microorganism to sleep away until where they get no return, where they get to a point of no return. So, and I've told you that it is the same epithelial cells that has also formed the gap junction. That has formed what is called the gap junction. So let's say this is two cells. Okay, two cells. Sorry, this is the nucleus. Let's say this is the nucleus of cell A, and this is the nucleus of cell B. So here it is the epithelial cell. This is exactly where it is called the tight junction. This place is where it is called the tight junction. And what happens with tight junction? It is this part of the epithelial cell that they will use to communicate, that cell A and cell B use in communication. So if there is infection of the O cell by microorganism, this cell A and B, they will communicate with each other. It's just like when armed robbers break into your house and you are armed with telephone to be able to call 911. So it is the epithelial cell that will serve as a medium of communication by providing what is called the tight junction between the epithelial cells. Okay, so the cell will crosstalk. We call them crosstalking. So this one will crosstalk, we talk with this other one, communicate with this other one. So for example, if the infection is coming from A, the epithelial cell here is going to send out communication to cell B so as to come to their rescue. Okay, now the next thing we will look at is I mentioned physical and chemical barrier. The physical and chemical barrier. Okay, so part of the chemical barrier I told you is the use of cilia. Sorry, the use of tears and saliva, and so on, in order to be able to prevent the microorganism from establishing itself. So don't forget that I said that the innate immune, they have the ability to be able to collaborate with the adaptive mechanism of the adaptive part of the host defense mechanism. So I have mentioned that of the barrier that is being put up by skin, whereby we consider epithelial cell as a major barrier junction that will serve as the first line of defense whenever the line of defense of the skin is already broken. So that means that will mean the internal organ will be so much at risk of infection. So don't forget that I said the primary aim of the host is for the host to prevent the establishment of infectious agents in the host system, whereas it is also the ultimate goal of the pathogen to make sure that the host is not competent enough, the host is malnourished, in order to be able to for them to be able to fight against whatever insult or mechanism that they might want to put up. So the arm robbers are pathogens, then the host, you are the landlord. So whatever you want to do to make sure that your environment is very very secure, or your house is very very secure. So some of them will do like by putting barbed wire, by having very thick gates for them to be able to break through. In fact, sometimes there is some people will also install alarm so that anybody that touches it is going to alarm the owner. So the alarm is the one that is being put up by the tight junction. Just like I said, that's what the host we use in communicating the order that already we have this. And don't forget that I told you that the epithelial cells also contain certain T lymphocytes. So that is one of the line of linkage between the innate immune system and that of the adaptive. And again, I told you that also the epithelial cells also contain some slippery agents or slippery leaf or to a point of no return. Okay. Now let's quickly go to the second aspect, which has to do with the second component of the innate immune system, which is mainly cellular. The cellular component of the innate immune system, cellular component. We have just mentioned 7. We mentioned 7. The natural killer cells. We mentioned eosinophils, basophils, macrophages, neutrophils, and dendritic cells. Okay, so dendritic cell. Okay, so we are going to start with the natural killer cells. Okay, what are they? The natural killer cells are the first. They are the first component of the innate immune system that we recognize. That there is an infection, a foreign antigen or foreign substance is already. So it is the first line of innate immune system to recognize. So that so it's just like when you are talking of the okay, it's just like when you are talking of gate man. You have a gate man. So the first person that we know that, oh, armed robbers are around should be your security agent. So the natural killer cells are the security agents. They are the security agents because they are the first to recognize They are the first to recognize. What do they recognize? They are the ones to recognize that certain cells are under stress. They are under stress. So that means if you are under any stress, your body, your immunological response will make sure that the natural killer cells are recruited. They are recruited. So they will recognize that the body is under stress. Not only that, not only recognition, they are also important because they are armed with different types of killing mechanism. So they recognize and kill. That's why they call them NKC, natural killer cells. Okay, so from natural killer cells is that they also contain on their surfaces. On the surface of natural killer cells, they have certain chemicals like glycoproteins. They contain glycoproteins, and what do these glycoproteins do? So this glycoprotein is the one they will use to recognize. They are the one that they use to recognize that there is a foreign body. There is foreign body. So apart from glycoproteins, they also contain some substances which are lectinized substances, which they use to activate what is called the classical pathway. What is called the classical pathway. We are going to see this when we get to more of this when we get to the complement system. But at this juncture, you should know that I said natural killer cells, they are the first to recognize the infection, that there is an infection. There is an infection, or the cell is already under stress. And whenever the cell is under stress, natural killer cells will be recruited to respond appropriately. And how would they respond appropriately? It's just because they contain glycoproteins. They contain glycoprotein. Don't forget, glyco from glucose and protein from protein. That means glucose, that is protein that contains glucose on them. So what do they normally use this one for? They use it for cell-to-cell communication, recognition, and they use it in what is called signal transduction as well. Signal transduction, because that's what they use to recognize. Then the natural killer cell is also lectinized. It contains lectin, and it is this lectin that will stimulate the classical pathway of the complement system. So we are going to see we are going to see it by the time we start talking about the complement system. But at this point, you should know that complement system consists of a classical pathway as well as a alternative pathway. So it is the lectin, it is this lectin by natural killer cells that will go and activate the classical pathway of the complement system, and we are going to see this just as we go. Then the natural killer cells, apart from recognizing, apart from killing, another very important thing that they do, that is, they have the ability to initiate inflammatory response. Inflammatory response. Inflammatory response is the swelling of certain parts of the cell. So and this inflammatory response, they are proteolytic. They are proteolytic in nature. That is, they are capable of breaking down or degrading the enzymes from microbial cells. So because it contains, and also this inflammatory response also has a kind of link with a complement pathway. We are going to see this as we go on. Then, apart from inflammatory response, another thing that they do is they will also trigger what is called opsonization. Natural killer cells have the ability to trigger opsonization. And what is the meaning of opsonization? Opsonization means that they are going to make that cell, the invading cells, they will make them to be palatable. They make them to be palatable for swallowing, for phagocytotic cell to be able to swallow them. So they will make them, they will prepare them. They will normally prepare them to be swallowed. So that is for in a process that is called opsonization. So we also see this when we also get into the complement system. When we get to the complement system. So that is why um, the next type of cellular components we are going to consider apart from natural killer cells is the mast cell. Number two, the mast cells. Okay. One very important thing about the mast cell is that they have the ability to secrete what is called histamine. They have the ability to secrete histamine, and histamine is a kind of a chemical that they use for anaphylactic reaction. Histamine for anaphylactic reaction. We are going to see more of this as we go on. But the mast cells, they are derived from bone marrow. They have their origin from bone marrow. So, and they have the characteristics. They have the characteristics of what is called cytoplasmic granules. They are made up of cytoplasmic granules. That is, granules that will make the invading cells, to be lysed. It will just go and lyse. It's just like when you have a dehydrating agent. So this cytoplasmic membrane of the mast cell contains liquid that dry off the invading object. So they contain cytoplasmic matrix that will dry off the invading microorganism. Then apart from that, the mast cell will also go and activate TLR. TLR means toll-like receptor. So natural mast cell will activate TLR receptor. So what do they make? What will the TLR receptor? What would they do? They will make the membrane. They will make the membrane of the invading cell to be permeable. It will make the membrane of the cell to be permeable. It will make them to be permeable to the host. So, and another thing is that it is the toll-like receptor. It is this mast cell that trigger. I told you that they are the one that triggers histamine. They are the one that triggers histamine, and histamine has to be allergic. It will make them to be allergic. So we are going to see more of this as we also go on. So, and this allergic condition is what is actually responsible for bringing out symptoms of infection, for bringing out symptoms like diarrhea and so on. So it is the mast cell. So anytime you see that you are already bringing out symptoms, don't forget that you can actually use symptoms to be able to diagnose that infection, to be able to diagnose. And what actually brings about that symptoms are what? The mast cells, because they trigger the secretion of histamine, and that histamine will bring out is the first response to allergic reaction, thereby bringing out symptoms of that infection. Because once you see the symptoms of that infection, definitely you are going to make sure that you treat it very well. You treat it very well. So that is about mast cell. That is that about a mast cell. So the next one is eosinophils. Eosinophils. Okay, the next cellular component. I told you that the first one is natural killer cells, and we have examined the basic function of natural killer cells. What do they contain? They contain glycoproteins. Apart from that, they contain proteolytic enzymes to be able to make sure and fight against the infection. Then we also look at the mast cell, which I told you that they are the one that creates histamine. And what will histamine do? The histamine will make sure that it triggers symptoms of that infection, so that the body will continue to put up that symptom so as to be able to. So the third one is eosinophils. Eosinophils. Okay, looking at the structure of eosinophils. If you look at the structure of eosinophil, you will see that eosinophils, it's what, whenever you collect blood sample, for example, when you collect blood sample, the next thing you see serum, white blood cells, and so on. So the eosinophils is a kind of okay, so eosinophils is a diagnostic. They use the percentage when you do total WBC. When you do total WBC, you will discover that WBC, different types, I think there are 5. You have basophil, which is an indication of infection by parasites. You also have eosinophils in particular now. So when, depending on the percentage of eosinophils in the body, that it is an indication of viral infection. So in case the invading microorganism is a virus, if it is a virus, it is going to trigger more of eosinophils to be secreted. And what do? They have also different mechanisms by which they respond to viral infection, such as making them to be able to go through lysogenic cycle instead of a lactic cycle. Don't forget that lactic cycle is what is going to make the virus to be able to bring out other, let me say, millions of viral particles. But lysogenic, that means they will not be able to, it will just be there, but it will not be able to divide. It's not going to be able to divide. So they also have what is called, eosinophils also have inflammatory action as well. They also have inflammatory action. So, and because they are extracellular, they have that ability to also trigger what is called inflammatory response. Next one, we have talked about natural killer cells, mast cells, eosinophils. So the fourth one is going to be the macrophages. Okay, I told you about that macrophages. They are very, very, very important. They are professional phagocytotic in nature. In fact, they have the ability to swallow anything. So once this one activates, it is eosinophil that activates macrophages. And whenever they are being activated, it will swallow microorganism. So a basic example of this is when you have latent TB. Latent TB. So what happens in latent TB is that the macrophages is very, very professional and is very, very active. So it's going to swallow the bacilli and prevent it from damaging, from further multiplication. What they do, I said, they swallow, that is, they ingest as well as also destroy. They have the ability to swallow, they have the ability to destroy, and apart from that, they swallow. They have the ability to swallow, they have the ability to digest, then they also have the ability to clear. They will clear the debris. They clear the debris without leaving any trace. So they swallow. So then apart from that, they also induce certain cytokines. They will induce certain cytokines. For example, they have the ability to induce cytokines like TNF alpha, as well as interferon gamma, IFN, TNF alpha, and interferon. So what would these cytokines, what do they do? What are cytokines? Cytokines are cyto. Whenever I told you that, whenever you come across the word cyto, that is talking about cellular. And kine is coming that it is a very small protein, so which are being triggered or recruited to the site of infection. And there are certain, for example, whenever you see this TNF alpha or interferon gamma, that is exactly what they use. Is like a biomarker. This one will serve as a biomarker or an indicator of an infection. So anytime you check, you collect sample and you see, if you are looking for how to diagnose latent TB, for example, the indication of interferon or interferon gamma is an indication of a latent infection. So all these principles, some of them are being used as a kind of a diagnostic mechanism. They use it as a diagnostic mechanism or diagnostic procedure for certain infection. So I told you that macrophages, they have the ability to swallow, to digest, and clear out the debris. Then number two, they have the ability to trigger, to induce the production of certain cytokines, and these cytokines will respond appropriately. I told you that certain cytokines will serve as biomarkers for diagnosis of police. You see them, you call them mobile police. Anyone that comes, they have the degree of their toxigenicity. They have the degree of preventing. So I told you that macrophages, they are professional phagocytotic in nature. They are professional. So, and that's the reason why that macrophages, there's a way, all these cellular components, there's a way all of them will cooperate with each other. There's a way mast cell will cooperate with natural killer cells, that we cooperate with eosinophils, macrophages, and so on, like that. Okay, I've told you that natural killer cells also stimulate the or trigger interferon gamma as well. So, and these are part of what we are going to use as diagnostic tool, indication for diagnostic tools as we also go on. So the next one after that is the neutrophils. So that is number 6. Neutrophils, just like from that word neutral, just from that word neutral, they are also professional. They are also professional um, phagocytotic, also in nature, that they are capable of natural, they are capable of what is called immunological surveillance. They have the ability for immunological surveillance. They have the ability for patrol. They will be moving around just like whenever you have alarm, or when you have alarmed the security that, oh, there is a different fluid of the body. So, and it is this neutrophil that is also called the PMN. The PMN is a polymorphonuclear lymphocytes. Polymorphonuclear leukocytes. So, and these, they are the most abundant in the blood, where you have them between 4 and 10,000 cells per ml of blood, between 4,000 and 10,000 cytokine response. So, responder, I told you, because they found themselves as undergoing normal patrol. So they are the early responder. If there is any infection, they are the one that will respond first. Apart from the natural killer cells that first of all recognizes. So the PMN or the neutrophils, they are the one that will first of all be the first responder. So the first one that will, so they are just like quick response uh, of first responder of the immune system. So unfortunately, the only thing is that they are short-lived. They do not live. They just exist within a within very few hours, and they die off. So, so the last one is the dendritic cells. The dendritic cells. So the dendritic cells is the last cellular component of the innate immune system. They are the last cellular component of the innate immune system. Don't forget that we have come across the component of the immune system, which I said it is the first one is going to be the barrier, and that barrier is going to be physical barrier and chemical barrier. The physical barrier, that is where we mention the skin and cilia. So, and we actually look at some of the reasons why the epithelial cells of the skin, they are keratinized and so on like that. So apart from that, the chemical components of the barrier, the chemical barrier of the innate immune system is where you have tears, saliva, and so on, as being the part of the physical barrier or chemical barrier of the immune system. Then I also mentioned the presence of hydrochloric acid in the stomach that raises the pH of the immune system so as to make them inhabitable for microorganisms. So we also mentioned the second component is the cellular component, and we have examined the natural killer cell, their structure, how do they look like. We talk about mast cells that is that is able to produce histamine for allergic reaction as well. We look at eosinophils, and after that the professional phagocytes, macrophages, as well as neutrophils. So the last one is going to be the dendritic cells. It's going to be the dendritic cell. So the function of the dendritic cell, that is called, they call them dendritic cell or DC. Anywhere you come across DC, they are referring to dendritic cells, and they perform majorly two major functions. Now, they serve as mediators. They serve as cellular mediators. They serve as cellular mediators between the innate immune system and the adaptive immune system. So they are just like an intermediary between the two arms of immune system. So they also have, apart from serving as intermediary, they also have the ability to stimulate adaptive immune response. So whenever the dendritic cells now sees that, oh, the innate immune system can no longer cope with the mechanism of the pathogen, so it is the dendritic cell that will link innate immune system and adaptive system together. Apart from linking, it is going to serve as the primary stimulator of the adaptive immune system, ability to produce inflammatory response. And don't forget, whenever there is an inflammatory response, generally there's going to be identification, or it can be used as source to diagnose infection. So that is I told you that dendritic cells serve as chemical mediators. They serve as they also serve as a bridge, a link between adaptive immune system, and innate immune system. Okay, so that is that about the cellular components of the innate immune system. So lastly, the last mention that today we are not going to spend time, because a different video will be dedicated for that. So, and that is the complements. That is the complement system. The complement system. Okay. Please find out that complement was actually discovered. Okay. Complements. The complement system. So just like the word complements, they are its labile, stable. They are its labile set of proteins that complement the efforts of macrophages. They complement the efforts of the innate immune system. Okay, I said this is discovered by certain scientists that it is its labile, and it's a very important component of the human blood plasma. This one is found in the blood plasma, and what do? They complement the phagocytotic efforts of the cellular components of the immune system, and that is where they derive their name. So then, apart from that, they also have complement system